Welcome to the Tech Lab C-Diff Quick Check Complete product demonstration. These materials are included in each test kit. Refer to the package insert for additional materials that may be required. First, add the diluent to a tube. Next, add 1 drop of conjugate. Then transfer the fecal specimen to the tube. Close the tube and mix the specimen thoroughly, either by vortexing or by repeatedly inverting the tube. Using a new graduated transfer pipette, transfer 500 microliters of the diluted specimen to the small sample well on the Quick Check device. Incubate for 15 minutes at room temperature. Add 300 microliters wash buffer to the large reaction window on the Quick Check device. Add 2 drops of substrate to the large reaction window. Incubate for 10 minutes at room temperature. Read the results promptly after 10 minutes. Refer to the C-Diff Quick Check Complete package insert for detailed interpretation guidelines.